Dear students, in the previous lesson we have already learned about granulocytes and agranulocytes, and we had a little bit of discussion regarding blood coagulation. Today in this lesson we are going to continue with this blood coagulation, right? So what did I say? Blood coagulation is a process in which the blood loses its fluidity, right? And as a result, the blood which is in liquid state will get converted into semi-solid substances, right? Now, this blood coagulation, as I've already said, that it is also known as blood clotting. So what did I say? Blood coagulation, this process initiates through two pathways. One is the extrinsic intrinsic pathway, another one is the extrinsic pathway. Fine. And this phoboblastic, what it does, in the presence of calcium ions, it results in the formation of prothrombinates. Okay? Now this prothrombinase, what it does, is an enzyme. So what is this? What is it happening? Thromboplastin, in the presence of calcium ions, it releases an enzyme known as prothrombinase. And these prothrombinase, in the presence of prothrombinase, what happens? The prothrombin, what it does, the prothrombin gets converted into thrombin. So in the presence of prothrombinase, prothrombin gets converted into thrombin. Fine. Now, in the presence of thrombin, what happens? Fibrinogen gets converted into fibrin monomer. Okay? Now, in the presence of thrombin, fibrinogen gets converted into fibrin monomer. Fine? Now, fibrin monomer, it gets polymerized. That means this fibrin monomer undergoes polymerization. If you write it here like this, I don't have space so much. Polymerization. If you can't follow this handwriting, please make sure it is polymerization. I'm written polymerization. Fine? So, fibrinogen is getting converted into fibrin monomer in the presence of thrombin. Now, this fibrin monomer, it undergoes polymerization to form fibrin polymer. And after it is converted to fibrin polymer, thread-like network appears, and this results in the formation of blood clot. This is the entire process that happens during blood coagulation. Fine. Now, our next subtopics will be blood group. Now, what did I say? Blood group. So now we are going to talk about blood group. Now, first and foremost, we need to know who has discovered this blood group. Okay? Scientist Pearl Landsteiner. Okay? Pearl Landsteiner, he first classified blood groups into four types, right? And he discovered it in around 1900. In 1900, Pearl Landsteiner classified this blood groups. He classified this into A, B, AB, and O. Okay. Now, before I discuss this in detail, you need to understand first what is antigen and what is antibody. After you understand these two terminologies, I will proceed further into the blood groups. Okay. What is in the blood, or rather in the same blood of a person. Listen to me carefully. Agglutinogen with its corresponding agglutinin cannot be present in the same blood of a person. What does that mean? next class. Okay? Till then,